Hello, everyone. Welcome back to the DIPG podcast. I'm your host, Dr. Marcelo Ortegal. This podcast is where we talk about DIPG and other pediatric brain tumors. The DIPG podcast is brought to you by the Mark Jr. Foundation. Phil Pan from California is our featured speaker, and we're going to discuss a number of things related to drug sensitivity tests, functional precision medicine, and things that Phil did while trying to extend or save his son Xavier's life after Xavier was diagnosed with DMG. Xavier was only 18 years old. He was diagnosed in 2020, and he passed away 4 years later. I think we're coming up on 2022.
22 months, so
we're coming up on 4 years since Xavier's death. And today is also a very special day for me, as I marked 7 years of losing my daughter to DIPG in 2019. Xavier led a very courageous battle against DMG, a 22-month battle altogether. Phil, like me, is a PhD scientist. He was trained, again, like me, as a molecular and cell biologist. I went towards immunology later on, and Phil went into toxicity and remained in molecular biology, and he has spent a lifetime in the biotech industry, in drug discovery as a scientist, and he used his skills to try to bring about a potential effective therapy for Xavier, which is incredible. And Phil's going to talk to us about this. Using his scientific background, he examined Xavier's biopsy cells against a panel of drugs called the drug sensitivity test, in where Phil will explain the details of how this was done. And essentially, when you run this test, you're asking what drugs will kill the tumor cells. Based on the results of this test, Phil went a step ahead. He actually recommended to the oncologists, the team of oncologists treating Xavier, what he thought would be the best course of action for Xavier's DMG. He developed a 2-drug combination in a very personalized medicine type of situation. We will discuss what that means. And Phil believes strongly that his approach extended Xavier's life significantly over potentially a year what other drugs might have done. Last year, Phil was the main author of a paper that was published in a peer-reviewed journal named Frontiers in Oncology. And Phil is a strong proponent of the approach that he took in terms of treating his own son, and that is functional precision medicine. He continues to promote functional precision medicine and to find personalized drug therapies for each of the DMG patients. And what Phil did for his son suffering from DMG is something that could also be applied to DIPG and to a variety of other brain tumors. With that, I welcome Phil Tan to the channel.
Thank you, Marcelo. Thank you for hosting me, especially on this day of Juliana's passing. I don't have to ask you how you're doing, but I admire what you're doing with the cause. I really express great gratitude for that. I don't know how you do it, because you're always doing it. And after I wrote that paper, kind of really in part to honor Xavier, right, and to try to get the word out and to hopefully help other patients. I had to take a break and kind of walk away from things. So I really admire what you and some others Yeah. So, as you said, Marcelo, yeah, I'm a PhD level biologist that worked in the pharmaceutical industry, and I developed a lot of experience analyzing drugs and how they work, and to be able to figure out which drugs work well and which drugs don't work just by looking at data. Yeah, and then when Xavier got diagnosed, like most of us, I knew about cancer, but I didn't know about DMG, DIPG, right? I'd never heard of it. And so I became, an amateur, maybe even a professional, developed professional knowledge and at least a professional scientific knowledge on DMG. Um, because there's very few treatments for it, maybe even professional clinical knowledge on it, to try to, desperately save Xavier's life. When he was diagnosed, I was, I don't have to tell everybody I was in shock, and I just signed all the papers that, authorized the people treating him to do all those tests, right? And I didn't even know what I was signing, but his, his, yeah, let me tell you about him first. That's a picture of him behind
Oh yeah,
me. It's,
please do. Please tell us about Xavier.
yeah,
It's
yeah,
important. I mean, our kids are not their diseases,
yeah.
and we sometimes have to remind ourselves that, and we have to find joy in the
Hmm.
time that we
That's right.
spend with our children, in the memories that we will carry for the rest of our lives, and we need to be always proud of talking about the kids, and because that's the way we move forward. And please do
Yeah.
so.
Thank you. Yeah. So he was 18 when he was diagnosed. So, he lived a complete childhood and actually had just finished a year in college. We live in San Diego. He was at University of California at Irvine. So he had moved away, um, not too far away. And it was 2020. So you remember what happened then. It was the year of, the beginning of COVID. And during that year, we saw before COVID, we saw him for the last time before COVID started at a volleyball tournament. He was a pretty high level volleyball player. It wasn't intercollegiate, but it was the club volleyball, and we went to see him, and he was doing so well. He was a starting outside hitter for the top team for uh, their club volleyball, and you know, uh, yeah, he was really enjoying being in college and being away from home as
And
college
for
students
those who
should
don't
be.
know, Irvine is about halfway between San Diego and Los Angeles. I know the area
Yeah,
very well. I used to travel up and down on the 405, and then the 5.
that's right.
But this UCI is.
It's about a 90-minute drive, which is, far enough, right? And you get away. When COVID happened, you remember a lot of students were sent back home. At his school, they gave him the choice of either going home or staying on campus. He lived on campus in a dormitory, and he wanted to stay on campus, and we let him. And Yeah, but he was one of the only students who stayed on campus. So that was in March, and so he had a few months of just being pretty isolated up there, really. And when he finished school in June, he came home. Right. He wasn't, he wasn't the same. We could tell he was showing the symptoms of the disease, and which was subtle at that time. Just kind of tired, and not very alert or active. And well, like most cases, we thought it was just well, it's the COVID isolation. We thought it was for those reasons. But long story short, we think it could have been diagnosed maybe a couple of months before it actually was that, the symptoms kept getting worse, and we kept, the physicians were looking at other causes, and they were focusing on constipation, which was pretty severe. And it's a common symptom of brain cancer, actually.
Right.
And, when he got diagnosed, his brain was, he was like, he was almost falling into a coma, actually. And his brain was experiencing a herniation from hydrocephalus, right? That's how bad it was. And finally did a scan and found the tumor, they told us that they had to do an emergency surgery right then and there just to relieve the hydrocephalus, right? Or else he might have died the next day. So that's where it was. Yeah. And of course, that's the day that changed everything, and things were never the same. But yeah, when he we were pretty isolated with COVID. We kind of were very careful and cautious, and when he passed, and we had the funeral, so many of his friends showed up, including a lot of his college friends who we had never met before, and we just found out how popular he was in college, how much he was adored, and how he just lit up a room, you know, um, and he just really blossomed up there, and you know, yeah, so yeah, that was heartbreaking, kind of, nice to see, but heartbreaking, of course. Yeah, so I guess getting back to his treatment journey, um, he had the usual so he had a thalamic DMG, not a DIPG. The thalamic DMGs are more common in adolescents, people like Xavier, and like most patients, his tumor was biopsied, right? And analyzed by molecular genetic profiling, which identifies the mutations in the tumor, and also might fully diagnose the tumor. So they'll typically look for the H3K27M mutation in probably any brain cancer, not just by looking at the DNA, but by doing pathology staining, and that confirmed the diagnosis. And based on that, a clinical trial was offered that was a personalized therapy that matched the mutations in his tumor, right? Besides the H3K27M mutation, there was a mutation in P53, which is also very common, and it's probably the second most. It's the most common mutation in all cancer. Mutation in PI3 kinase, and a lot of people know about that. That was the only mutation that was actually considered a targetable mutation, meaning there are drugs designed that specifically act on that particular protein. The drug that's being evaluated currently is Paxlovid for treating DMG. That drug back then was not available. It was still it was in clinical trials, but I think only in Australia. And so he got prescribed a number of drugs. One that kind of didn't target PI3 kinase directly, but something downstream in the same signal transduction pathway, which was an mTOR inhibitor called everolimus. And this drug has not been effective. He was also treated with panobinostat, which was a hot drug back then, from Michel Manji's lab. And they threw in temozolomide, which we know doesn't work for DMG, but the genetic profiling suggested that it might. That I think that was that was just an error in the genetic profiling, which can happen when they do this big omics analysis. And there was a fourth drug, hydroxychloroquine, which was intended to boost the effectiveness of everolimus. So he did radiation therapy and then this personalized precision medicine therapy. Precision medicine meaning based on the DNA sequencing of the tumor and targeting the mutations. And then that personalized therapy lasted for just 3 months because it was not working and the tumor grew back.
A quick reminder, pemvidostat is an HDAC inhibitor, and the TMZ has had at least conflicting results from the DIPG folks. I mean, because a lot of times people want to mix that with the radiotherapy, and some oncologists are vehemently against that mixture because of the side effects that it can generate. And some have seen some supposedly beneficial effects.
It's the standard of care for glioblastoma,
right?
Even though it doesn't work very well, right?
Right. And there's it can be extremely toxic, and that's again many of the problem.
And then, by the way, pemvidostat is no longer available, right? But back then it was an investigational drug. It actually received a conditional FDA approval, so it could be prescribed to anybody, but it was a conditional approval, and then the developer of that drug had to come up with some more data to get full approval.
Because it's very generic. It would hit all 6 different HDACs, ranging from HDAC 1 through HDAC 6.
Yeah.
And pemvidostat is very non-specific. It will hit all of them.
That's right.
Whereas it appears, if I'm not mistaken, HDAC 2 and 3 are the ones mostly associated with these types of tumors. So you'd have to find the deacetylase, a histone deacetylase inhibitor that is more specific, and they are available, which is just that was not known. So these are available, and folks watching the channel can discuss that with their medical team about the possibility of using one of those very specific drugs in combination with a kinase inhibitor, for example, or ONC201. We'll talk about that more often, or one of the imipridones. The imipridones are, as I mentioned before, the ONC series of drugs. But anyway, so please, Phil, continue telling about the story. And would you mind walking our viewers in terms of your screen? I mean, the paper that Phil wrote, and by the way, that paper will be linked into the description of this video cast, and you'll be able to read the paper. But Phil has a panel of roughly about 170 compounds, if I'm not mistaken, right, Phil? 170, 180
175, yeah.
175 compounds that he analyzed, looking for effect on Xavier's own tumor cells, and that is critical here in terms of this functional precision medicine, which is individualized based upon what the profile, what your tumor telling, or what tumor cells are telling the investigators in terms of what are they susceptible to, what are the drugs that will have the best effect using the least amount of drug, which Phil's going to mention that, which it has to do with the molarity or the concentration of the drug per certain volume. And the lower concentration, it typically is the best, because then you're likely to have fewer side effects, and you're not going to have the increased toxicity because you're using so much of that drug. There are a lot of nuances to that, and Phil will certainly dig into that and provide the viewers of the channel here some really good insights. Phil.
Yeah, thanks, Marcelo. So I should say that the paper I wrote, I'm not the only author, right? Of course, Xavier's a neuro-oncologist. He actually pushed it through, right? I forced it, right? I wrote it. I said, we got to get this out, right? When his neuro-oncologist, who is John Crawford, who was at Rady Children's Hospital in San Diego at the time and is currently at CHOC, the Children's Hospital in Orange County, which is close to Irvine. When he got on board with it, he kind of provided his clinical perspectives, and he submitted it to Frontiers on Oncology and got it in. There's three other people on the paper who are very important. There's Robert Wexler-Rea, who was a scientist, a really famous medulloblastoma researcher. I'll talk a lot about him because he's very involved in functional precision medicine. He's currently at Columbia University and was at the Sanford Burnham Travis Research Institute in San Diego at the time when Xavier was alive. There's Tim Martins, who does the drug screening up at the University of Washington. He's the guy that produces all this data. And finally, there's Pamela Becker, who kind of developed the concept of functional precision medicine, mainly for treating leukemia, and kind of developed this lab up at the University of Washington in collaboration with Tim. Yeah, and I had to have an affiliation for my authorship on the paper. I couldn't just say myself, or I was not employed at the time. And so the Cure Starts Now is my affiliation. They were great throughout Xavier's journey, and I continue to be active in raising money for them. And then also the Yuvan Tawari Foundation. So Parvati Tawari is yet another DMG parent who has been very active at supporting research in DMG. Yeah, so getting back to Xavier's journey, the precision medicine approach that I just described, based on targeting his DNA mutations in the tumor, did not work at all. And it turns out this has not worked in general for treating cancer patients. Maybe it will get better, but only if you look. I looked at a few large studies analyzing this, and it only benefits about 10% of all cancer patients. That's where it's at, and that's pretty bad, right? I think it will get better. It'll continue to get better.
So that shows an important discrepancy. Phil, excuse me for interrupting. Important discrepancy, because the precision medicine is based upon the association of the mutations that you will find by doing a genetic screening or RNA-seq analysis of the tumor. And then you have drugs or compounds that have been matched with these mutations.
That's right.
And then you would assume that having that mutation, a particular drug may be effective. And in Xavier case, Phil showed that is not necessarily the case. You have to look at the actual response of the tumor, of the cells, to whatever compound you're using. That is likely a more objective way of assessing a specific drug or combination of drugs that you want to hit the tumor with.
Yeah. Okay, so Xavier is 7 months into his journey. The clinical trial with the precision medicine, personalized therapy had failed. His tumor was in progression. It was growing again. He had to go back into surgery to receive an internal shunt, so he was in trouble. The tumor was about the size of an egg, it was pretty big. Back when he was diagnosed and when the biopsies were taken, and I was signing all those papers saying, yeah, do whatever testing you have, right? They did something called drug sensitivity testing, and that was through a research grant that from Robert Wexler-Rea, the medulloblastoma researcher that was collaborating with John Crawford, Xavier's neuro-oncologist. I didn't know anything about it, He also did just measuring some cell surface markers of the tumor cells, which is not discussed in the paper, but markers like PDL1, right? The HLA proteins. Another another protein called CD155, which is the poliovirus receptor. That one's pretty interesting, and they're doing some research on that too. But the drug sensitivity testing, right? The concept is, okay, when you take the biopsied tumor cells, you keep them alive, right? Most for most biopsies, the cells are killed, right? And then they're analyzed, right? The mutations are examined. There's pathology, right? But the point is the cells are dead, right? If you keep the cells alive, then you can just ask the cells, okay, what drugs kill you and what drugs don't kill you? Just ask the cells. It's simple, right? And it's the most straightforward approach to finding the most effective drugs, and also, just as importantly, identifying drugs that won't work at all, right? And the analyses like spit out some data that I could understand, right? One is the potency of each drug, right? That isn't how potently each drug killed the cells, right? The more potent, the more effective the drug is, right? The other measure is something called the efficacy, which is the percentage of cells that were killed at a high dose for each drug. So interestingly, some drugs can kill just, maybe half of the tumor cells, right? Other drugs will kill all of the tumor cells. Some drugs will kill none of them, right? So you want a drug from this, from the data from the screen, you want drugs that are highly potent and also kill most or all the cells. So they're both highly potent and highly effective, right? And I was able to take that data, and there was a drug that really stood out to me called disulfiram, right? Because it had those features, right? And also it's an FDA approved drug, right? It's been around for decades for treating alcoholism. It's a deterrent for alcoholics, um, and also it's cheap, right? Because it's FDA approved and it's been around forever. It's off patent and all that. You still need a prescription to use it. Um, and I just told Dr. Crawford to use it, and he didn't have any issues with that. To my knowledge, Xavier is the first DMG patient to use disulfiram, right? We didn't use it alone. We combined it with ONC201, and I kept asking about ONC201 because I knew that a couple of people with DMG had responded very well to it. I think at that time the publications were out, or at least the word was out. What's very interesting is that I was not I'm a pretty private person, and I was not into social media at the time, um, and so I didn't know about those Facebook sites that, like Jerry Ty's you know site, or the ONC201 sites. But I kept asking Dr. Crawford about ONC201, and
Just a quick addendum to our viewers, Phil. ONC201 and ONC206 that I mentioned earlier, they're a class of compounds called imipridones. And when they were discovered, they were classified new compounds. ONC201 is now the first drug approved by the FDA for pediatric DIPG. And it carries the name Modesto.
Dr. Crawford, he knew he can get it in Germany. He told me this. He said, if you search for it on the internet, I think you would find it. I realized he's talking about social media. So Xavier he's our youngest kid. He has two older siblings, and, of course, they're more savvy with social media. And so I asked them to help out, and, very quickly they found those Facebook sites about ONC201, and within a few weeks, I had ONC201 from Germany, and so we combined it. The drug sensitivity testing, interestingly, didn't really pick up ONC201. So ONC201 was not potent. If you just looked at if you just looked at the data alone, it wouldn't stand out. But I used it anyway because we knew more about it. We knew that it really benefited some patients, and that it also there was evidence that it stimulates both the immune system and the nervous system to help fight cancer, right? And these are features about drug sensitivity testing which are not captured. With drug sensitivity testing, just to kind of just try to describe the procedure in more detail, the biopsy tumor cells, at least the way it's done with Robert Wexler-Rea, they're actually dispersed. The tissue is dispersed and the cells are individualized, right? And then they're tested against the drugs. And so when you do that, you lose like the three-dimensional architecture of the tissue, and you lose a number of the features of the tumor, and you probably lose the immune system component as well. I knew ONC201, has some interesting features where it might stimulate the immune system to fight the tumor. And so, features that are not present in a drug sensitivity test. And so I decided to use it anyway. Very, very quickly, after this combo was started, the tumor shrank dramatically. And within 3 months, it shrank by like 80%.